Nitrosamine correlation to neurodegeneration, Lauren Sales. Nitrosamines and N-nitroso compounds are formed by chemical reactions between nitrites or nitrates and secondary amines or proteins. These function as both carcinogens and transplacental mutagens. Among more than 300 N-nitroso compounds were studied, and over 90% of them proved to be carcinogenic in various organs including the liver, GI tract, kidney, and bladder, just to name a few. All mammals are susceptible. The two main types are NDMA and NDEA, which are generated under strong acidic conditions like our stomach or increased temperatures like flame broiling or frying. We are exposed to these via multiple facets, the primary facet being nitrate-containing fertilizer, which is used across the country. These especially affect our root vegetables, like potatoes, as they contain 85% of our daily intake of nitrates per serving. This then leaches into our water supply, which is used for crop irrigation, food processing, and drinking. Then we go on to our tobacco, which nitrosamine is produced by the reaction of nitrite present already in the saliva with nicotine. This then turns into a tobacco alkaloid, which then progresses on into our nitrosamine. Our diet is also a very large precursor to nitrosamines, as fish byproducts, cheese products, beer, non-fat dried milk, and cured meats are all culprits. Cured meats especially, as sodium nitrate is used primarily to prevent toxin production as well as preserve color and flavor. But when you heat this, a nitrous acid formation occurs. So we start with our sodium nitrate, and with this high acidic environment or high temperatures, we end up with a nitrous acid. Moving on to this nitrosonium ion, which is a cation. This cation acts as our nitrosating agent, which then reacts with dimethylamine to generate our nitrosamine. Nitrosamines lead to the breaking down and destabilization of DNA, increased oxidative stress, lipid peroxidation, protein adduct formation, inflammation, and insulin resistance. So nitrosamines have these toxic and mutagenic effects by functioning as alkylating agents. Therefore, nitrosamines are converted during the metabolic process into active methylating agents that are highly reactive, especially with nucleic acids, which causes the DNA damage through gene expression. What's interesting is this is the exact same process as aging, which leads us to our Alzheimer's disease. Oxidative stress is the main precursor here for neurodegeneration. We start with our inflammatory response, aging, and this right here, which I want to highlight, is our peroxynitrate, which is an unstable structural isomer of nitrate, which then, as you know, leads to our nitrosamine. This is a precursor to our oxidative stress, which leads to either synaptic damage or apoptosis, leading to our Alzheimer's. In all of these studies, STZ is used. It is just a form of NDEA that can be isolated and used in bulk for studies. This first and second study were very similar, so we harvested the cerebella of 9 rat pups in both. The first was treated with NDEA in either a high-fat or low-fat diet for 8 weeks. In addition to the cerebella, the blood was used to analyze gene expression, insulin, and IGF. In the second, the rat pups were treated for 48 hours with strictly just NDEA, and only the cerebellar neuron cultures were accumulated. And again, instead of NDEA, it's actually STZ. The results for the high-fat diet with NDEA was neurodegeneration. They also examined the proceramide genes in the blood and found that they were upregulated, and ceramides cause insulin resistance and oxidative stress, which equals neurodegeneration. This proves that the mechanism stimulates the synthesis of ceramides and proposed other toxic lipids in the brain. The second study found that the NDEA impaired the mitochondrial function while increasing oxidative stress as well as DNA damage, which once again leads to our neurodegeneration. The third study was a human study. This was to test the metabolic pathways via high levels of sodium nitrite. The methods were through healthy non-smoking humans ages 22 to 30 years old. They took their blood and treated them with differing concentrations of sodium nitrite, performed their tests, and found an immediate loss of ATP and acetylcholinesterase, several indicators of oxidative stress, both lipid peroxidation and protein oxidation increased, an increased rigidity of cells which equaled susceptibility to lysis, and overall protein structure was altered. So here we have our Alzheimer's disease pathway. We're starting with our neuron. And what I specifically want to highlight here is that peroxynitrate, once again, leading to all of these consequences that we wanted to avoid earlier, including protein oxidation and mitochondrial dysfunction, which then brings us up to our mitochondrial dysfunction inside the mitochondria, which shows that ATP depletion, which we saw in the human study. All of these lead to our apoptosis or cell death, which ends up with that neurotoxicity and eventually neurodegeneration. So how do we fix it? We can regulate our fertilizer and irrigation methods, test water for nitrate content, ban nitrate fertilizers, reduce nitrite and nitrate preservative uses, especially in our diet, and cut down on smoking and availability of secondhand smoke to the public. Nitrosamines cause neurodegeneration. It has been proven and it can be avoided. That is the main point to take away from this, is that we are completely in control of how much this can affect us in our daily lives and affect our future. Please pause here for our citations. Thank you.